During the first stage of meiosis, interphase, the chromosomes of the cell duplicate to prepare for division of the cell. Interphase during meiosis is slightly different than during mitosis. One reason is that the centrosomes also duplicate. Also, only chromatin is visible and not the individual chromosomes. Like interphase, in mitosis, however, each chromosome still consists of two sister chromatids.
Prophase is the most intricate and time-consuming stage, taking up 90% of the time needed for meiosis to occur. During this phase, the nuclear envelope breaks down and chromatin coils up into chromosomes. This is also the stage in which homologous chromosomes pair together. This process is referred to as synapsis. Finally, in prophase, the centrosomes move away from each other and toward the other end of the cell.
Metaphase is the stage in which paired chromosomes align in the center of the cell. The spindles of the centrosomes then attach to the chromosomes in preparation for anaphase. One spindle, coming from one side of the cell, attaches to one homologous chromosome, while another spindle from the opposite end of the cell attaches to the other chromosome. Each chromosome is dense and compacted with sister chromatids fastened at the center.
Anaphase involves the chromosomes moving toward the ends of the cell. Even after moving, there are still two sister chromosomes attached to a centromere because the chromosomes will divide again during the second anaphase. This is achieved only by dividing the homologous chromosomes. These are known as tetrads.
When telophase begins, the chromosomes reach the cell's poles where haploid chromosomes are situated. However, all are still in duplicate form, containing two sister chromatids. Cytokinesis often transpires along with telophase, creating two daughter cells haploid in form. Within some life forms, the chromosomes then unwind, the nuclear envelope reforms, and a new interphase commences. In other species, daughter cells previously produced instantly prepare for a second life. This time around, the cell skips interphase and goes right into prophase 2. During this stage, there are half the amount of chromosomes per cell, and the pairing of chromosomes cannot be visualized. Other than that, it is extremely similar to prophase 1, except for a few events that only occur in prophase 1.
Throughout the duration of metaphase 2, the chromosomes align on the metaphase plate, like in mitosis. Moreover, the kinetic coils of each chromosome's sister chromatids point toward opposite poles. In addition, spindle fibers are affixed to the centromere of each homologous chromosome from both sides. Finally, the metaphase plane becomes perpendicular to that of the first metaphase.
As the second anaphase begins, the sister chromatid centromeres finally separate. Each pair, now identified as a distinct daughter chromosome, travels toward the cell's opposite poles. Both poles have an equal amount of chromosomes, but only possesses one chromatid. It is quite different from the first version of anaphase, where the homologous chromosomes are divided. She never stops, she's a go-getter. Takes him out to look.
During the second telophase, nuclei are developed at the cell's poles, as well as a nuclear envelope surrounding each chromosome set. The spindle apparatus vanishes, and much of the time, the chromosomes also endure decondensation. Meanwhile, cytokinesis takes place. Now, 4 daughter cells exist, each with a single set of chromosomes. As a result of crossing over, some chromosomes possess recombined parts of the parental cell's chromosomes,